Can people on Zoom hear me and see the black screen? Okay, awesome.
Let's assume they're fine so that people.
Okay. Great. Hi, everyone. Firstly, thank you so much for the opportunity. This is my first CogHear, and it's been super exciting so far. And thank you all for being here. So I'm Shailee. I'm a postdoc in Eddie Chang's lab at UCSF. And today I'll be talking about our work on modeling single neurons in the human brain during speech perception. So over the course of this fantastic workshop, we've heard a lot about different neural measurement techniques like electrocorticography, stereo EEG, MEG, EEG, and what have you. All of these neural measurements are fantastic, but they really tell us about what large-scale neural populations are doing in the brain during speech perception or production. But today I wanted to talk about a very different neural recording technology known as neuropixels. So neuropixels probes are these really thin wires. They're thinner than a strand of your hair. And unlike all of the modalities I just mentioned, they don't go on the surface of the cortex or the skull, but they actually go inside the cortical surface. So before some tissue has to be resected in a patient for epilepsy or for tumors, neurosurgeons, specifically Eddie, put the neuropixel inside, and this allows us to measure the firing patterns of single neurons across all layers of the cortex while our patients are listening to really simple English language sentences, which allows us to study speech at the level of single neurons in local neural populations. So just before I get started, I just wanted to give you a flavor of that data. So what you're going to be seeing here on the x-axis is time, and on the y-axis, you're going to be seeing the spiking activity of about 10,000 neurons that we've collected from a bunch of patients so far. On the top, I'll show you the auditory waveform as it's playing, and on the bottom, you'll see a summation of the population activity. So at every time point, I just add up the spikes of all of the neurons.
Does this bother you?
A flame would use up air.
12 o'clock level. There's no war on now.
Disturb her.
Okay, so you get the point. And I think, I hope you can appreciate just how exciting this data is in terms of the fact that for the first time, we're able to look at thousands of neurons in an awake human's brain while they're doing speech perception. And I want you to take away a couple of things from this. I think the first most striking thing about this visualization is how speech-locked the neural population is. This is, of course, not surprising. We're measuring from the superior temporal gyrus. We know STG loves speech, but you can really see in the population level code that as soon as speech happens from silence, there's an increase in spike, and then it goes down again and goes back up again. And the other thing to note here is how diverse the neural firing patterns are. I sorted the neurons based on the latency of their peak response. And you can see that there are neurons up here which fire basically all the time, but they vary in the firing rate. And there are neurons here which fire extremely sparsely. Some of them actually fire at the same point independent of which sentence you're in, which is a very interesting thing I want to talk about next. So this is just to give you a view of the data that we're working with. Oh, yeah. All of that was across people. It was about 35 patients aggregated. Yes, yeah, exactly. Yeah, so there's like some very interesting comments to be made about the consistency of neural firing patterns in organization across people. Yeah. Yes, so because they're across people, I haven't really ordered them based on anatomy or cortical depth. I only ordered them based on the latency of the peak response, such that the really sparse firing ones end up at the bottom and the really constant firing ones end up at the top. Yeah. So going to my actual title, this was sort of clickbait to make it seem like it's a BCI talk, but what I really want you to focus on, it was also logistical challenges, but what I really want you to focus on is we're working with this large-scale neuropixels data set, and we're going to be focusing on how these neurons work together to more understand how speech is encoded in these models than the traditional BCI decoding setup, but this is something that we're working towards. We ultimately want to be able to build these really nice self-supervised models of the neuropixels data, but this is not something I want to talk about today. It's more what we're working towards. Okay, so all of the results I'll be showing you today are from 17 patients, so just a subset of those 35 patients I showed you earlier, and they have coverage all across the superior temporal gyrus from anterior regions to more posterior regions. Now, a very fruitful and classical way to approach this problem is how we approach a lot of problems in speech neuroscience. You fit TRS, right? You can take the instantaneous pitch, you can take the instantaneous loudness, the MEL spectrogram, and you can build really effective interpretable linear encoding models. And this actually goes a long way. Laura and others have found really interesting single neuron encoding through this method. But what I was interested in is slightly rethinking the speech framework and trying to move away from these more instantaneous feature representations to something that can reflect how contextual and sequential speech is. So speech isn't really beads on a string. This is a metaphor we use all the time. It's a much more contextual, integrative process. And I'm curious about what role single neurons play in that contextual integration. So how do single neurons, for example, encode the time that is critical in speech? So how do they keep track of temporal order of information? How do they integrate contextual information? What sorts of representations do they learn? Those are the broad types of questions my work is trying to answer. So in order to answer these questions, I turned to another system that is also extremely effective at continuously, contextually, and sequentially representing speech, but it's not the human brain. This is the third talk of the day, so you don't even need to guess. It's an artificial neural network. And this is just one neural network. You can put in your favorite one. It doesn't matter. But the point is that speech DNNs have gotten really good at representing contextual integrative information, right? Like Jean-Remy illustrated this really nicely. Sam's work illustrates this really nicely. And so what my work is trying to do is try to draw comparisons between these deep neural networks and the single neuron populations we're recording with NeuroPixels. There are many, many different ways you can approach this problem. In today's talk, I'll be talking about two different ways. The first way is based on traditional encoding models. This is very similar to what you heard Jean-Remy talk about and Sam talk about, wherein you take the latent or internal states of these deep neural networks, and then you use a linear encoding setup to predict the spiking activity of either individual neurons or the population. And then you can do a lot of really fun interpretability things to now understand the artificial neural network, and as a consequence, understand the linear predictions it's making about the brain. So just to be clear, the paradigm is that you take the hidden states of these deep neural networks, use them to predict brain activity, then you insert interpretability prompts into this artificial neural network as a way to understand something about the brain predictions. So that's the first angle that we're going for. There's another angle that I'll touch upon at the end, which doesn't really look at these predictive models, but looks more at the representational geometry of artificial neural networks and neural populations in the STG. And what we're trying to answer there is, are the representation manifolds the same? So do these artificial and biological systems represent phonetic sounds in the same structure or word boundaries in the same structure? But that is towards the end of the talk. So before I actually tell you what sorts of really interesting interpretability we can do with these models, just to quickly tell you how well these models work, this is very similar to a graph you just saw Sam present. On the x-axis is the percentage of variance explained in a test set with acoustic linguistic features, and on the y-axis is a deep neural network. Again, doesn't matter which one. Every dot here is a neuron, and you can see that an overwhelming number of neurons are extremely well predicted with deep neural networks by almost sometimes by 10 to 20% more. But this is honestly not surprising anymore. We've known this for 6 years now. Instead of fMRI or ECOG, I'm showing you this in NeuroPixels. So speech DNNs are incredibly effective at predicting neural spiking activity, but again, unsurprising. The more interesting question is, why do they work so well, and what can they tell us about the brain, right? So to answer this question, let's go back to the initial questions we had and think about speech as a continuous sequential contextual process. So what I try to do is I try to understand how these speech DNNs are achieving this context integration and then trying to link that to what the neurons could be doing. Specifically, I try to focus on interpretable computational motifs that exist in the deep neural network architecture and trying to see if they're also represented in the human brain. So just to explain some of those interpretability ideas, I'm going to lay them out here and then go into each of them in detail. This is a very classical architecture for modern deep neural networks. This is a transformer if you haven't seen it. Transformers are what all of the fancy models are now made of. But as complex as transformers are, they also have a lot of really cool interpretable units. And there's a lot of amazing AI work showing that you can pull apart different parts of these models to understand about how they're working. So that's basically what I did. So the first interpretable motif that I focused on was the position encoding in a transformer. If you're familiar with recurrent systems like LSTMs or recurrent neural networks, you know that these models operate sequentially, so they have an implicit representation of time. The number of non-linearities you stack on can tell you how much time has passed, but models like transformers don't have any recurrence, they just take in the input as a whole, so they have no idea about the order or temporal structure of their input. So to keep track of time, they use these explicit things known as position encodings. These are just fancy sinusoidal waves. And the way they work is on the x-axis is time. The y-axis are a bunch of different sinusoids with different periods. So this has a really short period, this has a really long period. And by just stacking all of them together, at any one time point, the model can look at the different phases of the sinusoids and infer where it is in time. And this is a really clean and easy way of explicitly representing time information that recurrent systems don't do. So we can try to see if there is such explicit representation of time in the single neurons as well, or they do something more like a recurrent system wherein the time is implicit but not signaled by any individual neuron. Another thing that you can do is to look at these, increasing contextual receptive fields. So this very much aligns with the two previous talks you saw today. Deep neural networks normally contain multiple layers, and as you go deeper in the model, the receptive field or the contextual field increases. And you can look at both how the amount of context fed into the model affects the prediction of any individual neuron, and which layer in the deep neural network that neuron likes as a clue into how much information it could be integrating over. So that's another interpretability technique. The third interpretability technique focuses on how transformers or deep neural networks achieve information integration. So transformers use this thing known as attention to figure out how to combine information across time. So just to give you an example, if you had a sentence, I actually can't read the sentence, but if you had a bunch of sentences here and you wanted to figure out which pronoun the word it was, which noun the word it was referring to, this is a classical co-reference resolution problem, what the model would have to do is it would have to go back in time and figure out what it needs to attend to, right? So it would have to go back in time, learn to attend to the subject, and that's the only way the model will know what the current pronoun refers to. And this operation of going back in time and attending on what is useful is known as attention. And a lot of cool AI interpretability work has found these attention units in transformers are extremely interpretable. The transformer I was working with has 144 such attention heads, each of which learn extremely different things about speech. Some of them like syllable boundaries, some of them learn word onsets, some of them learn biphone sequences. And by interpreting those attention heads, you can link to what operations the single neurons could be doing. So that's idea 3. All of these ideas that I've presented so far rely on encoding models for single neurons, but there's a very, very different way we can compare artificial neural networks and biological systems, and this has been largely ignored in speech neuroscience, but this idea is by comparing the representational geometries directly. So on the one hand, an artificial neural network is a combination of single neurons. The SDG is a combination of single neurons. So what you can do is look at how the population geometry of both of these systems varies and what sorts of factors organize information across both of these populations, how they differ, how they align. Does that explain why layer 9 in UBERT does well, for example? All of those interesting questions. So that is the last part of my talk. Okay, so to get into it, let's first talk about the time stuff. Specifically, the question we're asking here again is that, is there an explicit representation of time in single neurons of the human speech cortex? Now, obviously, I wouldn't be telling you this if I hadn't found it, but you actually do see neurons like this. So here on the x-axis is time from the onset of a sentence up to the end. And on the y-axis are 100 different sentences. I've ordered them such that the shortest sentence is at the top and the longest sentence is at the bottom. And this is the firing of one such neuron. So this neuron very clearly loves the onset of speech, right? So when you go from silence to a sentence, it really loves to fire. And when you model this neuron, you actually figured out that that position information, the position encoding, is enough to predict the spiking activity of the neuron. You don't even need the fancy latent states of a deep neural network, and the acoustic linguistic features don't do that well. But what's interesting is that we have 10,000 neurons. It would be extremely challenging to look at their spike rasters individually, but you can use this position encoding prediction framework to find out such example neurons. Here is another one. This neuron loves time around 1 second. So you can see that it doesn't fire that much, and then it really fires in the middle of the sentence and starts to taper off. And this is another neuron which fires towards 1.5 seconds to 2 seconds. And I think what's really exciting about this is that this is the first time we've seen explicit time cells in the human speech cortex. This is something that people have found in the hippocampus, for example, but we haven't really seen it in the cortex. We haven't really seen it for speech. And I think another interesting thing is that there's also some parallels with the hippocampus in that the hippocampal time cells tend to have time fields which go broader and broader as more time passes. So onsets are very sharp, but as you go on in time, they broaden more. And so that is an interesting finding. Sorry, I'm just going to.
I'm gonna go first.
Okay, yeah.
So,
Yeah.
why do you know it's just a random set of features that you're picking up in random projections and in time and frequency? Some are gonna be early and some are gonna be late because it's random features.
Oh, so I mean, we're not just taking features here, right? We're looking at the spike rasters of individual neurons, and the point is that independent of which sentence they're in, they always fire at a specific time. There's another question about whether these neurons are epiphenomenal, right? Like there could be that the population is the time readouts just happen to be an epiphenomena as a consequence of whatever these neurons are doing. Or it could be that there's a traveling wave and that's what you're picking up on. So something that we're still working on is trying to causally prove that the time cells are critical for the speech cortex to maintain time and are not just a readout because of something else happening.
Yeah, this is a good question. I mean, is this time relative sense concept or time relative to sound concept?
So we haven't actually, we don't have non-speech stimuli to verify against, but that would be interesting. Yes, there's a lot of silence.
So just be like how the delay or something like that.
Oh yeah, so we looked at whether it's actually wait, sorry, if it's a delayed response, again that relates to the epiphenomena question. Like, is it a delayed response, or is it that the timing, the 600 milliseconds is causally relevant to binding features at that time? So we're seeing some evidence for that, but it's been, we're still working on it. But yeah, it could be either of those.
So I'm really puzzled about these data sets. So you take the same sentence and repeat it 10 times, and you change amplitude to see whether or not you really have a robust timing, or, I mean, this whole thing could be artifact, right?
Artifact of what?
Well, it's just dependent upon the variability of a given cell at a given time. I'm saying, how consistent is it in terms of?
Oh, but these are 100 different sentences.
These are all the same sentence?
No, these are 100 different sentences with extremely different speech or acoustic features at any given time point, right?
Right, but let's say the sentence ends at different times.
Yes.
So there could be modulation of the phrase of the sentence. I understand, is it the timing of the sentence or is it the timing of the cell?
Oh, I see. Okay, it's not the timing of the sentence. So sorry, there's two different things that could be happening here, right? One is that there's the timing of the sentence, more like the structure yoke thing that Sam was talking about, wherein it's the relative boundaries of the linguistic units that's driving the firing of a neuron. We don't find evidence for that. What we find evidence for is very similar to what Sam was saying, wherein it's just locking to the timing, like the neuron just likes a specific time. It doesn't care about what is happening, what rate it's happening at, but it's firing at 600 milliseconds.
Okay. I mean, I'm just not convinced that, because you should be making the claim that it's specifically a time cell, but it may be a sentence cell.
Oh, yeah, yes. Yeah. So it could be, yes, okay. So that's a fair point. It could be a sentence cell in that I think what's important to note here is that it's encoding some specificity in terms of how much of the sequence has passed. And it could be specific to speech, it could be specific to like something about the sentence structure. But when you look at a lot of variability in acoustics, you can look at a lot of variability in the speech content, you still see this sharp response relative to the onset.
Just a clarification of what features are going into the speech DNN's temporal precision and the acoustic and linguistic?
Okay, so the acoustic linguistic is all of the standard TRFs like pitch, loudness, stress, smell, all phonemic categories, phonetic categories, all of those. The speech DNNs is I took all of the deep layers of the deep neural network from layer 1 to 12, allowed it to pick whichever layer it likes. And that is often as good as, or maybe slightly worse than taking just the position encoding alone. So if you take just the sinusoidal position encodings, which can only predict time, those position encodings are content invariant. And even if you have content invariant input features in your encoding model, it predicts well because it only cares about the time. Yeah. Yes.
Literally goes all the way to the sentence end, but it's all stopped short, like 50 milliseconds or milliseconds. It's like predicts the end of the sentence.
Yeah, it's actually it's not actually predicting the end of the sentence. It's just responding to the drop off in the envelope.
Decide the beginning and end, is it some threshold over the envelope or?
Oh yeah, so the sentence onset is the annotation. It's annotated to the start of the first phoneme or word. So that's what the sentence onset is.
Stop example starts at 0.
Yes.
There is none.
Yes.
But if it's a vowel, start high, so you can vary.
Yeah, and we also track the rate of change of the envelope. Yeah.
So I guess the same question. Then the ending can also be ending on a silence.
Yes, yeah, exactly. So it could just be it's like a gradual tapering off. Yeah.
And you get nothing back. Is that?
Typical or
it's not displaying?
I'm just not showing it. You do get stuff, but it's the firing rate is much lower. Yeah. Around 500 milliseconds. Which I think for single neurons is enough for them to reset and go back. So just to look at these time cells across a population. So here, again, x-axis is again time, but y-axis is a bunch of these cells that are stacked that are stacked on top of each other. I'm looking at 35 of these at a time. And the darker the pink, that is where the maximal firing rate is. And keeping the same order for this particular sentence, I can look at 2 other sentences. So the y-axis is now the same. And I think what's interesting here is that it's just illustrating the same point in that a neuron that likes around 600 milliseconds likes that time across all of the sentences. And also, this is all single child data and neuropixel. So it's quite noisy. So it's still pretty interesting that we're seeing some stability. And I think the other thing that stands out to me is the fact that you get this styling of time from the onset of speech up to, say, 2 seconds in, which is our longest stimulus. So you see single neurons responding to all of the different time pockets available for this task. So these are our first type of computational motifs that we've found. Great question. So we were really hoping to find some anatomical or depth localization, but what we've found so far is that these time cells are pretty broadly distributed across STG, and they often are highly intermixed with neurons that care about different types of speech features. So which again really motivates that analysis about what role they're playing in binding different features. But we don't see, it's not that they're only in posterior STG, they're pretty broadly throughout. Yeah, what percentage? Yeah. It's around 5 to 10% of all the neurons we have, which is, I think, pretty sizable. Yeah.
Just a quick interrupt. I want to ask a question, get my attention up in the microphone over. We don't absolutely need it in this room, but it definitely helps people online. So I'll keep an eye out.
Yeah, so I'm really intrigued by this cell. It's cool, but have you tried doing, compression and expansion to see whether or not the timing changes in the timeline?
Yeah, so we didn't go back, manipulate the duration and check, which would be a very cool experiment to do. But I've done a simpler version of that in that I checked wherein certain speech occurrences that have different rates or durations, like it's the same utterance but different durations, do the time cells vary? And they don't vary that much, which is why I think that they probably care more about absolute time than the relative structure. There are neurons that care about relative structure, so they do exist, but they're much rarer.
Oh, so you have structure cells and cells.
Yeah, but the structure cells are so rare that we still need to make sure that they're structure cells. We do see some cells that pattern that way, but they're nowhere near 5 to 10% of the data. Yeah. Okay, in the interest of time, I'm going to skip this next section, but I'll just tell you what we found. We also found these combination cells wherein they cared about time, but only when a certain acoustic feature was present or a phonetic feature was present. So we found, for example, nasal neurons that love nasals, but only at specific time instances in the sentence. So they don't respond to every nasal, they respond to nasals at a specific delay, essentially. So there are these combination of pure time cells or in time plus content cells. And then the third category, which I will jump to, which are these more contextual cells. So these are neurons that don't really care about when something happens. They care about what happens and in the context of what that thing is. So they care about a lot of contextual information. So there are many ways you can identify these contextual neurons. For example, the experiment that Sam was presenting is one such way wherein you manipulate the context and you see if the responses change, if the correlation changes. Another way that I developed during my PhD was by doing a very simple experiment wherein you manipulate the context available to the deep neural network, and then you evaluate wherein the additional context helps your encoding model. So essentially, you build one model that takes in no context. It takes in only the current time step. Another model that takes in, say, 200 milliseconds of context, and a third model that takes 400 milliseconds of context. So you can build these ranges of models that vary in the amount of contextual information they have, and you can evaluate whether neurons care about that additional context. Are they utilizing that additional context to be able to predict them well? And what we actually find is very similar to the results you've seen today, but at the level of single neurons. So here on the x-axis, I'm showing the increase in the variance explained with additional context. This is the histogram, so the y-axis is number of neurons. And I have loosely grouped neurons into phonetic and lexical stress. Now, these TRFs are not great, so you cannot say with confidence that these are stress neurons or phonetic neurons, but that is the feature in a traditional TRF that these neurons best responded to. And what's interesting is that there are these neurons that respond to these classical TRF features but don't really care about context. But there are also a long tail of neurons that show an enormous improvement in context when you add information beyond the level of the word they're in. So lexical level context and beyond really helps the predictive performance of a bunch of these neurons, which suggests that there's varying degrees of context sensitivity across single neurons in the speech cortex. So I will get in about 5 minutes. I'll tell you about what roles at the population level these different contextual groups play. But just to unpack this contextual integration a little bit, what we next did was to look at the mechanism in the deep neural networks that are responsible for context integration. And this is the attention heads I was telling you about. So what you're looking at is an attention map that is very standard for a transformer. Here, x-axis is the sequence and y-axis is time. So a bright pink indicates that at that particular time, the model was only attending to whatever is in pink. So for example, you can see that this particular attention head, it really only cares about local information, right? So it's extremely diagonal. It only cares about what happened immediately before or after it. It doesn't really care about anything that came much back in the future. And so you can classify these different attention heads and you can come up with very, very different patterns. For example, these are our local or diagonal heads that cared about the preceding or succeeding context. there were these more onset sensitive heads that cared about the diagonal structure, but they were also really sensitive to whatever happened at the beginnings. They were kind of latching on to the beginning and interpreting the rest of the sequence based on that. And then there's the third category, which is everything else that we didn't understand. These were extremely complex attention patterns. They didn't follow a local diagonal structure or an onset structure. We then looked at these different attention heads and we tried to see which of these heads best predicts single neurons. And overwhelmingly, we saw that single neurons are very well predicted by these local attention heads. So these are the number of well-modeled neurons on the y-axis. X-axis is the individual attention heads. And you can see that most neurons in the STG really care about local sequential context integration. I'm skipping this in the interest of time, but I can give you a one-liner. We then went inside these attention heads and looked at what their actual patterns were, like do they care about future context, do they care about the past, how far back in the past, do they latch onto syllable boundaries, et cetera, and different neurons liked different patterns. So there were some neurons that were almost peeking in the future. They were looking at whatever was going, like predictive coding. There were some neurons that cared about the boundary of the syllable way back when. There were other neurons that cared about the biphone structure, and we were able to figure that out by looking at these attention heads and looking at what the heads themselves latch onto. And what's important to note is that these attention heads actually have comparable performance to those high dimensional internal representations of deep neural networks. Those representations are like 768 or 1024 dimensions. Attention heads are usually just 64 dimensions. So having these relatively low dimensional representations is enough to explain single neurons really well. Yeah.
Any distribution correctly in terms of these?
Yes, yeah. So there is some distribution cortically. We are seeing some effects wherein a lot of the neurons that care about longer linguistic structures like words and syllables tend to be more anterior. But this is something we're also still working on because you have to account for SNR differences in the neuropixels and the dropout and things like that. So anatomy is a bit hard to prove, but we do have some evidence for anatomical organization for the temporal scale.
So when you're talking about all this, okay. When you're talking about whether something predicts the brain, predicts a neuron, you're always controlling for what? The spectrogram and these other features? So
Yeah.
it's all unique variants.
Exactly.
Above and beyond.
Yeah.
And do you have something that can like capture, maybe I can't tell if it's something you want to find or control for, but do you have something that can capture like onset responses and stuff like that, and just kind of low level, like additional low level stuff?
Yeah, a great question. Thank you. So I don't mention this, but all of the analyses I show you, we always control for like low-level features like onsets by having an explicit onset response, but the position encodings are also really great for that. And we also control for like the Mel spectrogram, envelope, all of the traditional low-level features. So anything that we say is well-modeled above and beyond those features significantly. Yeah, because like one thing that's kind of we didn't talk about is that there are several neurons that are well modeled even by traditional TRF. So it's not that the deep neural networks are always amazing, but they're overwhelmingly amazing, which I think is an important distinction. Okay, so I don't know how much. Okay, I guess I have some time. So just to go back to my title slide, because I have fancy words that make it seem like we're doing this like large scale thing, I want to now switch focus from single neurons to looking at population level codes and to analyze what the population level structure is. Also, I think I miscalculated and I may have more time than I thought I did, but so this section was going to be short, but I can keep talking. Okay, so I showed you these context insensitive and context sensitive neurons that we identified by manipulating the context available to the deep neural network. So the next question I was interested in is what role do these context sensitive neurons play in the population level code? Are they able to keep track of some information that the context insensitive group is not able to? So we had a very simple speech reconstruction experiment wherein we tried to linearly decode the speech signal from the context-sensitive and context-insensitive populations, but importantly, we did it at different temporal lags. So we analyzed how far back in time can you accurately reconstruct speech from the two different neural populations. And what you find is a very clear difference. So here I'm going to be showing you decoding results from context-insensitive neurons, context-sensitive neurons in the full population. And I controlled these two different groups for everything else. So they have the same SNR, they have the same standard TRF selectivity, firing rates, all of the things. So the first thing to see here on the x-axis is the temporal, the decoding delay. So this is decoding speech 300 milliseconds in the past, and the y-axis is the decoding accuracy. You can already see that the context-insensitive group is slightly worse, although I don't think it's significantly worse. But as you go farther and farther back in time, 500 milliseconds, 700 milliseconds, 900 milliseconds, you start to see that the context-insensitive group quickly decays in its ability to decode speech or to reconstruct the speech signal, but the context-sensitive group does not. So the context-sensitive group is able to very well predict the speech signal, even 700 milliseconds in the past. Now, obviously, speech is a highly autocorrelated signal. There's a lot of structure that is shared from what I'm saying right now to what I said 700 milliseconds ago. But what's interesting to note is that there's a difference in the population. So depending on how context sensitive these neurons were, they can or cannot tell you exactly what happened 700 or 900 milliseconds in the past, which is multiple words in our data set. And so these context-sensitive neural populations can reconstruct content at the scale of words, which is pretty interesting given the fact that these are just single neurons in the superior temporal gyrus. We actually also have some interesting work mapping the context sensitivity of ECOG and local neural populations. I can give you a one-liner and go in more detail if we have more time. But what we essentially find is if you look above, so we're recording from neuropixels, so we have a bunch of neurons in a putative column, and you have the ECOG electrodes sitting on top. So when you look at the temporal integration window or the context selectivity of the ECOG electrode on top, it's highly correlated to what you find in the population, but it's always longer. So even though these models don't differ it's not that the ECOG data is predicted better or it has higher SNR, controlling for all of that, you still find that the ECOG global response has a longer time scale than the underlying neural population, but it always patterns the same, which is a pretty, I think it's a very interesting finding to think about how to position all of the neuropixels work in the context of what we've already found with intracranial recordings.
Say longer by how much longer?
By several hundred milliseconds, actually. So from the, maybe, okay, maybe like a word or a word and a half. So, and it always happens. You always get that. So there could be many explanations for this. It could just be the way where, the neural measurement that you're picking up on from ECOG compared to the neuropixels, but it could also be that in traditional recurrence systems, for example, when you have units in an RNN that have very different temporal scales, by virtue of combining their temporal scales, RNNs can approximate much longer temporal structures. And so that's something that could be happening here, and we're working on circuit models to figure that out. Okay. So just one last thing, and I can end in time for questions. So the last thing I want to talk about is everything I've spoken about has been in this more traditional encoding, decoding framework, but this is very much in progress. So I won't actually show you the neural results because we're still working on them, but I was just excited to get everybody's feedback and to tell you about it. This is work I'm doing in collaboration with Chaim Sompolinsky at Harvard and his student Shane. And what we're trying to look at here is to try to directly compare the population geometry of biological and artificial systems. So a very typical way that neuro AI has progressed in speech and language neuroscience is by building these encoding, decoding models between artificial networks and the brain. But with the neuropixels data, we have this exciting opportunity to look at how these single neuron populations are structured both in a deep neural network and the superior temporal gyrus. So that's exactly what we're doing. So just to give you a sense of the types of measurements we're working with, Suppose you were trying to look at phoneme categorization, right? Like you want to know how does the human brain or a deep neural network categorize phonemes? How are phonemes represented in the population manifold? You could measure things like the signal or the dimensionality of that manifold. The signal essentially means how far are, for example, the fricative representations from the plosive representations. You can imagine blue is fricative and green is plosive, and you want to see how far apart are these two manifolds. The farther apart they are, the easier it is for the system to distinguish between them. Another thing that you can measure is how high dimensional are these representations, because the dimensionality can indicate the types of variances that system is using to classify between these phonemic categories, for example. So we're looking at a lot of these traditional geometric measures to look at how the population geometries differ. I won't show you neural results, but I will show you deep neural network results. So here on the x-axis are the layers of a state-of-the-art speech model. This is one of the whispers. It does really well. And on the y-axis are the two different measures. One is the signal-to-noise ratio, and the other one is the dimensionality for phonemic manifolds. And what's interesting here is that as you go deeper into the model, specifically in the upper middle layers, that's when the signal is really high for phonemic separation, but you also get a ramp up in dimensionality. And the reason I think this is exciting is because in some preliminary analyses we've done, we're kind of seeing a very similar effect in the phonemic manifolds of the neuropixels data, but it's different across anatomical locations. So we're able to see a higher separability in anterior sides compared to posterior from the preliminary data that we've worked with. But I also think that this is an important way of looking at these deep neural networks, because this increase in signal and dimensionality can even tell us why those parts of the DNN predict brain data well, right? So you saw through multiple talks now that these upper middle layers tend to predict very specific parts of the brain well, but we don't really know why. And I think the fact that these phonemic manifolds are so separable is one clue to that question. And another clue, which I didn't talk about in the attention section, is that those local sequential integration attention heads are also much more abundant in these layers. So we're trying to converge on a story about how the computational mechanism, which is the attention, the manifold, which is the signal separation dimensionality, and how these artificial neural networks are able to do phoneme separation, and what that can tell us about the superior temporal gyrus. So just to conclude, I think maybe the key takeaways from what I presented to you today was that there are these single neurons that show sensitivity to both time and content beyond just feature detection. So there are not single neurons that are like, okay, high pitch sound or high loudness or whatever. They actually care about these much more complex structures like time or contextual integration. While prior theories have suggested that a lot of this is beyond the means of STG, for example, a very standard Hickok and Popel model would suggest that a lot of this happens outside of the STG. STG does more phonemic level stuff. We actually see a lot of sensitivity, even at the level of single neurons, to word level structures and syllabic structures. And the fact that you're seeing these explicit time representations and these time plus content cells is making us really rethink how speech could be organized, even within the superior temporal gyrus. Because if it was just a pure recurrent system, you may not need these explicit time representations. So I would love to hear all of your thoughts about how the system could be organized given the types of single neurons that we're finding. So with that, I would like to thank you all and to thank my collaborators and specifically my postdoc advisor, Eddie. Thank you.
Thank you. Just, can you give us a sense for which of the things that you found you kind of can't see at the macro electrode level? Like what stuff, like what new stuff can you see at the level of single neurons that you couldn't see at the macro?
Okay, that's a good question. So I think a lot of the different types of like descriptions we've come up for encoding, you often do see at the ECOG level, but they're just either they're more mixed or yeah, so they're usually much more mixed, I think. So you can see, for example, some hints of temporal coding in the ECOG electrodes, but they're often extremely mixed and you don't find pure temporal structures, you often find time plus something else. So I think that is one thing. You see a lot of much more heterogeneity in the response. And the other thing that I think could be useful to look at the level of neuropixels is, because I guess it's an underlying question, is this a useful representation level to look at for speech, right? Like, do you even need to look at the level of single neurons? I think that's an interesting and compelling question. But I think the fact that we're seeing these very different types of representation profiles could maybe enable us to look at local circuits in a way that you simply can't with intracranial recordings, because they're just so heterogeneous in terms of what we capture.
I'm so excited about this stuff. I think it's so cool. So like I was very inspired by Jim DiCarlo's work. And one thing that's cool there is you can decode people's ability to recognize objects at levels that are comparable to what a human can do. And I've never seen anything like that in geometry. I've never seen a single, plot that showed you could decode anything at human levels of performance. And presumably it's because our measures are crappy, but you have the real thing. So yeah, I'm curious, can you decode anything at a human level of performance with big population?
Yeah, I haven't benchmarked to that yet, but yeah, I agree, that would be exciting. We are able to decode a bunch of things almost as well as the entire ECOG population, like the whole grid, but it would be nice to be able to benchmark that. Thank you.
Any other questions for lunch? Oh.
Okay, yeah, I don't know what Neil's question is too, but I really am intrigued by your results. I think they're really cool. I'm just trying to get my head around them because it's a little bit puzzling to me as to what your results really are with the timing specifically. So a couple of little questions. So for the stimuli you're presenting, the sentences you're presenting, if you change the length of a sentence, do the timing cells, they're invariant?
Yeah,
So have you actually done that kind of thing at all? Or are you essentially looking at a set of different sentences? And then, making the claim that for all the sentences that you present, a given cell still responds at the same time.
Yeah, so we did a slightly different version of what you're asking, wherein I ordered the neurons. I ordered the latent neurons based on their latency using short sentences. So I grouped the sentences into short, medium, long based on duration. And I order based on short, and I evaluate how well the order persists in the long sentences, and it still persists well. So which means to say that if there is an ordering of latency across the neural population for extremely short stimuli, when you present really long stimuli, that relative structure still persists. Does that? Yeah, and another important criteria in identifying a time neuron was to explicitly check for this. So we very tightly control the variability we allow in the peak latency across sentences. And only if that variability is low do we show it.
And you're using neuropixels, so is there any differential in the depth, the laminar position of these cells?
Yeah, great question. I would have loved to answer that question, but it's been slightly challenging because across different recordings, you are not guaranteed to get the same number of, equal distribution of neurons across depth, and they also have lots of SNR differences and dropout. So that's been very challenging to control across the data set, but yeah, it would be a very important question.
So what role do you think these cells are actually playing? I mean, I guess that's sort of the other question. I mean, they exist in 5% of the cells that seem to have something like this timing characteristic. But in terms of the question of how are they useful for decoding a given sentence, is there something about, are they more commonly found at a phrase level? I mean, have you analyzed that at all? Yeah, I think we've done the first step, which, like you said, is to identify them. Something that we're still working on and is not clear is what they're doing, which has been a harder question to answer. I can tell you some possibilities we're exploring. So one possibility is that these time cells are locking on to a specific feature and are signaling to another area that now combines time and feature, right? Like these time cells often co-locate with neurons that like specific phonetic categories, that like stress, whatever. So it could be that perhaps they're talking to each other and then time locking
the feature representation. So that is one possibility. Another possibility is that they could be used to detect the boundaries of linguistic content. That is something I'm seeing less evidence for. At the ECOG level, we see very clear responses to, say, word boundaries. So you see a really beautiful high gamma dip when there is a word boundary, but that's been really hard to find at the level of single neurons. You don't see boundary effects in single neurons. And so I'm leaning towards the time plus feature tagging, but it's still something we're exploring. Yeah, thanks.
So 10,000 neurons, is not one. So 10,000 neurons sounds really amazing, but there's also a danger that if you had 10,000 completely random neurons,
Yeah.
you can probably still find all of these patterns in those, right? So how
Yeah.
do you make sure that this is?
Yeah, that's a good question. So it's like correcting for multiple comparisons, right? Like if you look at a broad enough thing, you'll find something. So the way that so there's multiple ways that we've thought about that problem. The first technique is the fact that even from those 10,000 neurons, if you remove some of them due to SNR, dropout, whatever, you end up with 5 to 10% of your data that very cleanly looks like this. And another thing that we're doing, which I didn't show here, is we're trying to see if these if you see time neurons in different types of speech inputs. So for example, everything I showed you here was single sentences, but if you had more continuous speech, how far can you stretch these temporal fields? Like if you had somebody listening to a story in the OR, do you now get time cells that tile the entire duration? So I think the way we're trying to see how generalizable this is by presenting more types of the same stimuli and more types of stimuli. And I think that might be a good way of establishing whether this is false discovery. But so far, it has worked. So far, we have been able to show robust effects that generalize well. But yeah, I agree it would be a challenge.
Thank you.